You're welcome to the third class of the Noble King Maths Bioinformatics beginner course. So, in the last class, we talked about assessing the databases, and this is the third class whereby we'll be doing some kind of a practical exploration of what these databases are all about. We've made mention of the two major classification, the ones that are talking about the nucleotide and the other parts that are talking about the proteins. So now I'll talk about two of the nucleotide database and one of the protein database and how you can interpolate between those materials. So the first one here is the NCBI. So Googling the just NCBI, you can get to the website, but because I've been used to using this website before, so it was easy for me to get to that. So that's not the point. So but you searching for NCBI on Google will take you directly to the page. Now this is how the page looks like. So, and there are different resources that you can find on the NCBI database. Now this is just the home page, sort of. This is my own home page. There are different home pages based on what you want it to do. But okay, let's take it step by step. So when you get here, you'll be taken to you'll see different interfaces whereby you see submit your sequences. If you have your sequences, DNA sequences you want to submit to NCBI, this is where you go. But you want to transfer NCBI data to your computer. Maybe you want to download a particular set of sequences. This is the download part. The learn part, the develop is self-explanatory. The analyze, that is identify an NCBI tool for your data analysis task. Is also a rich resources. Maybe you want to design a primer. This is where you search for the tool that you are going to use to design the primer. And then to search the research, explain CBI research and collaborative projects. This is also a rich avenue. This part is also a rich avenue for you to explore and do some research. Now let us be specific in what we want to do. The NCBI performs different tasks. So what you want to do will determine what we want to use this NCBI to do. Now, if you want to search for materials on this NCBI, so you can simply come to PubMed. PubMed is a collection of different materials or different papers, different research, and then the bookshelf, the PubMed Central, and then the almighty blast. So let us open the link in new tab, and then when you want to talk discuss about the nucleotide, the genome, the single nucleotide but this is, let us take it little by little. So let us go to PubMed. PubMed comprises more than 32 million citations for biomedical literature from Medline Life Science journals and online books. So basically, you can use a repository of different works of different research that have been done on biomedical literature. Just like we've said in the first class, that the goal of bioinformatics is to understand the information in biological data, like make a sense of all the ACGT, what does it really mean? What does this data, what does it really mean? So here is a repository of journals, of publications, of high impact publications that are used, that have been done for different analysis. Now, as you can see, there are different journals. We have Nature, we have Plus One, we have Pediatric Science, Journal of Neuroscience, Diabetes, Cancer Research, and the likes. These are high impact factor journals that have their publications here. Okay, let's search. For example, we want to know about coronavirus, COVID-19. Okay, let's do coronavirus, COVID-19. Let's search. So you can get a lot of papers that have been published. As you can see, the coronavirus publication is, let's say from 2003, you can see the traction. When you go to 2020 or 2019, that is when the highest number of publication of coronavirus was made. Although coronavirus is not limited to just COVID, we also have different coronaviruses that affect bats and other stuff that are not as pathological and pathogenic as and debilitating as COVID-19. So these are information. So as you can see, these are 2020 publication. You can show more. As you can see, we have more than 100,000 results. So it's a rich avenue for you to explore published information. Okay, let us check about malaria. 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 So you can, oh, sorry, double I. So you can see a lot of malaria publication. Of course, malaria is one of the most researched disease. You can see it, a lot of them here, a lot of them here. You can see a lot of whatever search you want to make. That is that about. That's just the tip of the iceberg for PubMed. Now we've opened Blast. Now Blast. Now okay, let me go. This is the Blast part. I've opened the tab. This is Blast. So Blast is the basic local alignment search tool. That is the full meaning of Blast. So what Blast basically does is find similarities between biological sequences. Like now, let's say I have a sequence of this particular bacteria. I have the sequence of Cardinium species, or Hamiltonella, or this Potera. These are bacteria, uh, the sequence. So what I would do is that if I want to find the similarity of my sequences with published data, we made mention of publications that have been made in databases, submitted in databases, and this is also an attempt to give a meaning to all this submission. Now the first thing you do is just come to nucleotide blast. At least you want to compare your nucleotide sequences with other nucleotide sequences. That is when you come here, you click that, and then the platform for you to submit opens up. I don't know what is wrong with my network. It is so slow, even though it is uh, I fast running. So here you submit either your accession number or FASTA sequences. That is when you submit your sequences, and then you blast. After submitting, just come here to blast straight up. So let me try to get some